I'm Janet Rossant. I'm a senior scientist at the Hospital for Sick Children in Toronto. I'm also president of the Gairdner Foundation. My research is really all about the early embryo, mostly working with the mouse. And we're interested in early development because during the early stages of development, we can derive different kinds of stem cells, including the famous pluripotent stem cells. So when you think about stem cell research and certainly human embryo research, there have been many, many different controversies over the years. But in human stem cell research, the biggest controversy really came about when people first derived human pluripotent stem cells. Did we predict it would be controversial? Absolutely yes, because we knew that in the mouse, we can derive from a mouse blastocyst, a very, very early stage of development when the embryo is the size of a speck of dust. It has a few cells. From that stage that we can derive these pluripotent stem cells in the mouse, and that we could show in the mouse that they give rise to every cell type in the body. In fact, we can make a whole mouse from pluripotent stem cells. As soon as you know that, and you know you can do it in the mouse, it immediately became obvious to, I think, all of us working in the field, that first of all, it would be incredibly powerful to be able to do the same thing in humans, because you could have a stem cell that maybe in the future you could use to repair every cell type in the body. But it was also apparent that if those stem cells were being derived from human embryos, then it was going to be controversial, because where are those embryos going to come from? And that, as soon as the first human embryonic stem cells were generated in 1998 by Jamie Thompson, it was a big story and a big ethical story. Those stem cells were derived from embryos that were left over after in vitro fertilization. So in reproductive technologies, when people want to have babies by artificial reproduction, by in vitro fertilization, usually more eggs and embryos are made than are put back. Those embryos are frozen away. They can be used sometimes for another round of reproduction, but often, at the end of the day, people have their babies, the embryos stay in the freezer. So with full informed consent, Jamie had been able to access some of these frozen embryos. And for many people, that's an acceptable situation. As soon as you know that there are going to be embryos left over, isn't it more appropriate to use those embryos to make cells that could help other people, rather than leave them in the freezer where eventually they will be discarded? But for other groups, the concept of just destroying a human embryo at all, destroying it to make a cell line, is unacceptable. For the Catholic Church, for example, life begins at conception, egg and sperm, and we cannot, should not ever damage that embryo in any way, destroy its potential to make life. Very controversial, involved with a lot of pro-life, right-to-life groups, very different all over the world, different countries having different concepts about this. But did we predict it? Sure. Can you argue against it? There were many arguments at the time and continuing today. This is still, I think, a big ethical concern. Well, today we're very concerned. The world is a lot of discussion groups around the possibility of editing the human embryo, and particularly what we call the germline. In other words, making changes during the development of the embryo that could be inherited to the next generation. This is certainly something we're going to have to grapple with, because right now the technologies are not very efficient. We're not really in a situation where we could do that. But as we move forward, where do we draw the line between preventing serious genetic disease, which could be done with gene editing, and human enhancement with gene editing? And that is something that we are going to have to grapple with. Where do humans think the boundaries should lie in our ability to change our own genome? That is certainly one. Sort of related to that, technologies that will allow us to change human reproduction and essentially generate human embryos in a petri dish, either by mixing stem cells together or taking stem cells, letting them change and differentiate in the dish, and making eggs and sperm from stem cell lines. This can be done in the mouse, and again, it's only a matter of time and technology to translate that into human. What do we feel about making eggs and sperm, making embryos from stem cells, not using normal human reproduction at all? How do we control that? What cells should we use? Who gets the opportunity to make embryos? And of course, initially, how safe is it? There are lots of issues before we could ever do this. But again, if one thing I've learned over my years in research, particularly the last 10, 15 years, technologies move incredibly fast, and things that we thought were impossible, like germline editing in humans, are now here. And so what we consider for the future as impossible, I don't know what's impossible. So to me, stem cell research, its initiation and its future really lies in regenerative medicine. I do believe that most of us started working on particularly pluripotent stem cells. Yes, they're interesting. We want to know where they come from. As a developmental biologist, I find them fascinating. But we really want to use them to treat patients. And I think the combination, in fact, of gene editing and stem cell therapy is going to be incredibly important for a number of diseases, and more diseases perhaps than we know about now. And I think, to me, if we can see diseases like type 1 diabetes, instead of people sitting on insulin forever, that we can cure it. So it's one and done. If you can make islet cells for type 1 diabetes, have them implanted in a patient, and have them survive. There are still a lot of questions along that way, but that's the goal. And stem cell therapy really is the kind of therapy that can actually be curative, and that's incredibly powerful.